Hi, I'm Dial Holton, Microcalorimetry Product Manager at TA Instruments. I'd like to welcome you and thank you for joining us today for this webinar. During the webinar, you can customize your viewing interface by arranging windows to your liking. You can resize the video and slide windows independently from one another by clicking and dragging the bottom right corner of each window. If you get disconnected at any time, please use the instructions you received to log back in. You can access several different content windows by clicking the widgets at the bottom of the screen. These include speaker bios, additional file downloads, and social media interactions. If you need help, click the question mark widget. Please ask any questions you might have at any time during this presentation by submitting them through the Q&A window. We will answer as many of these as possible at the end of the webinar. With that, I'd like to introduce John Ladbury. He was initially trained as a physical inorganic chemist at the University of London and the University of Greenwich in the UK. Dr. Ladbury completed his postdoctoral research at Yale University in biophysics, Harvard University Medical School in structural biology, and the New York University Medical Center in molecular and cellular biology. Dr. Ladbury was privileged to obtain a Research Career Development Fellowship from the Wellcome Trust in 1994. Which enabled him to return to the UK to start his own laboratory at the University of Oxford. After joining the University College London in 1996, he became a Wellcome Trust Senior Research Fellow, and in 1998 then became a Professor of Molecular Biophysics and successfully renewed his fellowship. In 2008, he took up a position of the Edward Rotan Distinguished Professor of Cancer Research and the Director of the Center of Biomolecular Structure and Function at MD Anderson Cancer Center. He has published seminal investigations on the characterization of water molecules and ions in biomolecular interfaces, and later he exploited this strength in biophysical chemistry in looking at protein interactions in intracellular signal transduction. His current research involves a multi-technique approach in investigating the ways in which proteins involved with signal transduction work in mammalian cells. Dr. Ladbury has published over 125 peer-reviewed papers and articles and has edited 2 authoritative books on biocalorimetry and 1 on structural-based drug design. He is a former editor of the Journal of Molecular Biology and currently a deputy chair of the Biochemical Journal. The title of his talk today is there some added value in measuring thermodynamic parameters in the drug development process? And with that, I'll turn it over to John.
Thank you very much, Dial. So today I'm going to speak to you about the use of thermodynamic parameterization in the drug development process, and particularly the idea of using measurements from isothermal titration calorimetry to input information into understanding a biological interaction and how that biological interaction can be used to make decisions on drug compounds. The entire talk is going to focus on the SH2 domain interaction, and you can see a representation of an SH2 domain in the slide before you. The SH2 domain was a major target for most pharmaceutical companies in the 1990s, since it plays a key role in signal transduction pathways, which when they are aberrant, give rise to disease states such as cancer, diabetes, and autoimmune disease. The image you can see in front of you is an image of an SH2 domain with a peptide bound. The SH2 domain has a canonical structure of a beta sheet region sandwiched between 2 alpha helices, which make 2 binding pockets, as you'll see on the next slide. The cognate ligand, which is represented as a stick model in this particular image, binds across the beta sheet region, and 2 of the side chains of the peptide that you can see here delve into 2 deep pockets, and hence this provided a structure which was extremely appealing to the drug development scientists. The 2 regions of the binding, you can see in the pink ovals on the image, correspond to a phosphotyrosine, which is on the bottom right, and in this case an isoleucine side chain, which is in the pink oval on the top left. If you look at the image of the surface of the SH2 domain structure, you can see very nicely how these two deep pockets provided recognition sites for the phosphotyrosine moiety on the right-hand side and the isoleucine moiety on the left-hand side. These two deep pockets corresponded to something like a two-pronged plug interaction, which enabled tight binding and recognition of the oncoming molecule. And hence the idea that you could design a compound which would optimize binding in those two pockets provided an interesting and largely unfruitful, as you'll find out, challenge. So you're seeing here a peptide represented by the stick model binding to the surface of the SH2 domain. And as I said, the phosphotyrosine moiety is absolutely crucial in binding, in as much as that's absolutely required for SH2 domain ligand interactions. Going from the phosphotyrosine at the N-terminal in a C-terminal direction, which is going from right to left, you see a glutamate and a second glutamate which splay across that beta sheet region, which I showed you in the previous slide, and then the isoleucine, which is in the phosphotyrosine +3 position, delving into the second pocket. So we're going to look at these interactions and look at how we can use thermodynamics to understand how chemical moieties can probe these binding sites. And the first site we're going to see or look at is the optimization of chemical moieties binding into the py+3 pocket. And you can see that highlighted in the yellow oval on the left-hand side of the current slide. There was a huge interest, as I said, in developing compounds that would recognize these SH2 domains. And I'm going to talk to you about one particular example and show you how thermodynamic parameterization could provide an enhanced level of understanding of binding of compounds which would potentially be lead compounds in the drug development process. In the image you can see in front of you, you can see a template for a series of compounds which were developed by a pharmaceutical company, which optimized the idea of a peptidomimetic, which would, as the name suggests, correspond to the peptide which you saw binding in the previous slide. The peptidomimetic template that you can see here has a phosphotyrosine moiety, which is in the bottom left-hand corner of the slide, and then a peptidomimetic backbone, which is rigidified by the dicyclic group you can see, and then a series of different chemical moieties were put onto the what assimilates the position of that isoleucine molecule, side chain or residue that you saw in the previous slide, which would be placed in the locality of the yellow circle you can see in this slide. So the idea was that a series of compounds were made with different chemical moieties which would have the phosphotyrosine moiety, which would bind into the phosphotyrosine binding pocket, obviously, and then a second chemical moiety which would explore that py+3 pocket. So the series of compounds, and I'm taking just a selection of 6 to exemplify this particular idea, were quite diverse, and you can see that on the right-hand side of the slide in front of you, a series of chemical moieties that were slotted into that yellow circle to make lead compounds. And these chemical moieties range from, as you can see at the top, cyclic aromatic systems, going down to aliphatic acid compounds, which clearly are quite diverse in their nature and would provide a significant difference in the way that they might recognize that py+3 pocket, always remembering that the other part of the molecule was maintained the same. So the point about this slide is to show you that when you actually measure the affinity of binding of these 6 different compounds to the SH2 domain, you see that, certainly from the free energy terms that are shown here, that the affinities of these interactions are essentially identical. Going for the KDs of these interactions are all in the micromolar to tenths of micromolar region, and the KD doesn't vary by more than an order of magnitude, which is extremely surprising based on the difference that you see, the variability that you see in these chemical moieties that are put into the supposedly exploring this py+3 pocket. But the point here is that you can make these measurements, and if you want to make a decision as to which of these lead compounds would be a suitable one to pursue in terms of developing a drug, you wouldn't really get an awful lot of information from these free energy terms. So the question then becomes, is there any benefit in measuring the thermodynamic parameters of these interactions? And in the next slide, you can see that when we look at the underlying enthalpy and entropy terms, you actually do start to see clear signatures and differences between the way that these compounds recognize the SH2 domain binding site. So although they have very similar affinities, the enthalpy and entropy terms contribute in a different way to that free energy term. And yet I can highlight that by looking at, for example, the first value there where the enthalpy term is something like minus 27 kilojoules per mole, and the entropy term is plus 7.2, which is clearly different from the molecule represented by the data at the bottom of this list, where the delta H is minus 16 and the T delta S is plus 19.3. So the point being here is that making the measurements using isothermal titration calorimetry highlights the different way that these compounds are recognizing the SH2 domain, even though the affinities are quite similar. So the big question then becomes so what is the point? Is there any real benefit in getting this underlying or second layer of thermodynamic information in terms of the making decision as to which compound might be worth taking forward? So what I want to try and sell to you, if you like, at this point is that indeed having this level of information can enable you to make a decision as to which compound would be worth pursuing. And the way that this can be done is by focusing on the enthalpic contribution of the binding as a way of being able to assess whether the compound is making a good complement of non-covalent interactions in the binding site. And to give you some more detail on that, the enthalpic contribution to binding can be a good parameter for compound selection, amongst, I should say, other parameters. But the point here is that if you have if you look at a protein-ligand interaction, the affinity will be improved by having an increase in the number and strength of non-covalent bonds, and that's fairly obvious. And since the enthalpy is giving you a readout associated with which is basically telling you the net change in non-covalent interactions, then by having a compound which binds with a more favorable enthalpy term compared to its entropy term, then you're essentially getting a good idea that compound is making a good complement of non-covalent bonds in the interface, as opposed to a system which is, for example, entropy-driven, whereby clearly the major contributor of that is going to be based on changing of degrees of freedom of the entire system. So the idea here is that by getting a favorable enthalpic contribution, you are, so if you take two compounds and one has a more favorable enthalpic contribution than the other, it's likely that compound which has the more favorable enthalpic contribution has a better complement of non-covalent interactions. And since the major contribution to enthalpy is going to be from hydrogen bonds, you can largely assume that compound is making a good complement of hydrogen bonds in the interface. And furthermore, since it's very difficult to design in hydrogen bonds de novo, to be able to screen and come up with compounds which have this already existing complement of hydrogen bonds in the interface, then you're in a good position to then build out from that molecule and improve the affinity in many cases simply by increasing the entropic contribution. Indeed, that's for the most part what medicinal chemists are extremely good at. So this then provides the idea that if we look at our series of compounds taken from a high throughput or medium throughput screen, and then look at the hits and establish an enthalpic contribution for those, we can use that enthalpy term to make decisions as to which ones to take forward. And the term that I like to sort of proffer as a way of as a decision-making tool is this idea of enthalpic efficiency, which is simply the delta H that you measured divided by a term n, which depending on how you like it, can be the number of non-hydrogen bond atoms, rather, or the molecular weight. And this is also clearly related to the usually adopted term of ligand efficiency. But in this case, clearly you're just, rather than using an affinity term, you're using an enthalpic term to assess whether you have a good contribution from the net making or breaking of non-covalent interactions. So clearly then, a compound with a higher or better enthalpic efficiency will be the one that you would ideally take forward to the next level of drug development. And the point I made earlier with respect to it being actually very hard to design in from a rational basis new hydrogen bonds is extremely important in this case because, as I said, it's actually easier to build out your molecule and gain increase in affinity from the entropic contribution of binding. And you can see that in this image here, whereby when you prior to bringing two molecules together to form an interface, the surfaces are largely hydrated, and there's interactions with those two molecules from the bulk solvent water. When you form the interaction, that water gets pushed out of the interface, and hydrogen bonds can form between the interacting moieties. In some cases, you get trapped water molecules, as you can see on the image on the right-hand side of the equilibrium there. And I'll come back to those later on in the talk. But for the most part, those molecules are excluded from the interface, pushed out from being ordered on the surface, and provide an entropic contribution to binding. And so when you add in an additional piece of surface area, as you can see in this version of the previous slide, I've just increased the surface area by building out at the bottom of the image on the right-hand side. A water molecule will then get pushed out and provide further entropic contribution to binding. So the bottom line here then is the enthalpic efficiency of a molecule can be used as a decision maker in taking a compound through the development process. So with respect to the 6 compounds that I showed you earlier, which were binders or based on that template, which you can now see in the image in front of you, the big question then becomes, well, does it really work? And so the different R groups that you saw, ranging from those cyclic aromatic systems through to the acid aliphatic groups, give rise to clearly different thermodynamic parameters, as you saw before. And those details can be seen in the slide in front of you now. In the green box are the different side chains that were put onto that template with the phosphotyrosine moiety, and the data that you can see corresponds to isothermal titration calorimetric data for the binding of these systems. I've also included in this table the enthalpic efficiency and indeed the ligand efficiency, which, as I say, is the commonly used term by medicinal chemists to rate the free energy with the size of the molecules that they're developing. So when we look at these data, you can see quite clearly that the compound 1 has the most favorable enthalpic efficiency, and I've circled those data with the red oval shapes. The worst enthalpic efficiency belongs to the aliphatic acid system, which is molecule 6, which are also highlighted at the bottom of the table. So this actually conjures up an interesting question, or at least an interesting answer or phenomenon based on those chemical moieties that were placed onto that template. In as much as molecule 1 has this cyclic aromatic group, which clearly is not a provider of hydrogen bonds, and yet it has the most favorable enthalpic efficiency. And based on what I've said to you, would be the molecule that I would suggest to my medicinal chemist friends to take on and develop further. And the big question is, well, where is this additional enthalpic contribution coming from? And I think we can reconcile that by looking in further detail at the structure of the SH2 domain, again, in this case with the peptide bound. And you can see here two representations of the SH2 domain. You can see the beta sheet region and the two alpha helices that I pointed out in an earlier slide. And in this case, our sort of cognate peptide, which has the phosphotyrosine and the two glutamates and the isoleucine lying across the beta sheet region, and that's represented in the black stick figure. On the right, I've just included the surfaces that the phosphotyrosine and the isoleucine would bind to, and you can see that on the left-hand side, the figure shows that the isoleucine is delving into a hydrophobic pocket, which is coloured in green. So when we plant the drug or the lead compound onto this, the phosphotyrosine moiety is going to bind in the same site, and the chemical moiety, the cyclic aromatic system, is going to reach over to that hydrophobic pocket and delve into there. So where does the additional non-covalent bonding arise from? So when you look in more detail at the binding pocket, which is shown on the right-hand side figure of the SH2 domain, you can see quite clearly there are two tyrosine residues which reach out from that pocket. And the idea here is that aromatic system from the lead compound gets sandwiched between those tyrosine and forms a pi stacking arrangement, and it's that which is providing you that additional enthalpic contribution. So I think one of the points to take home from this little exercise, and certainly in looking at these six molecules, is that you don't necessarily have to have very clear hydrogen bonding forming to provide an enthalpic contribution. It can come from other sources, such as, in this case, pi stacking of aromatic systems. But nonetheless, by choosing this molecule, you have a system whereby you have the most favourable arrangement of non-covalent interactions already established, and then to build up your molecule further, you're in a much stronger position than having to design in de novo non-covalent interactions. So moving on now, I'd like to sort of look at interactions in the phosphotyrosine binding pocket, which is highlighted in yellow on this surface representation. So the reason I want to delve into this particular area of investigation is that we were looking at finding compounds that would be able to replace the phosphotyrosine moiety as a part of an inhibitory molecule. And quite clearly, the reason for this is that the phosphotyrosine itself is hydrolyzable and so doesn't provide a very good part of a ligand. But furthermore, and which is actually more important for what I'm going to say to you today, is ideally the pocket that the phosphotyrosine delves into is, as you'll see in the next slide, is extremely has a large number of potential hydrogen bonding arrangements that it can make. And so the idea was to try and find other chemical moieties that might be able to exploit that pocket better than the phosphotyrosine moiety itself. And as you'll see, that's would be a major challenge. And the way that this was going to be done was by using taking a fragment library and which are low molecular weight chemical moieties, and test them against this SH2 domain and see if we could identify small low molecular weight fragments that would be better binders than the phosphotyrosine moiety. So as I said to you, the phosphotyrosine itself is in a pocket which offers a huge number of hydrogen bonds, and you can see that represented in the figure that you're seeing before you now. In the yellow oval is the phosphotyrosine moiety itself coming from the peptide or the chemical compound that would be the inhibitor to the SH2 domain. And surrounding that are the amino acids which form the pocket. And you can see, based on the orange dotted lines, the number of hydrogen bonds that are reaching out, or at least extending to the phosphotyrosine. And I think there's something like 11 hydrogen bonds that the phosphotyrosine is able to make. So it's extremely well complementary, or it makes extremely complementary interaction in that phosphotyrosine binding pocket. So as I said, it would be a major challenge to come up with a molecule that can do better than the phosphotyrosine moiety itself. And indeed, if you look at the energetics, which again are established through isothermal titration calorimetry, you can see that the phosphotyrosine itself binds fairly weakly. The binding constant here is 1.7 times 10 to the 3 molar to the minus 1, which corresponds to a free energy change of about 21 kilojoules per mole. So that in itself is not a particularly tight interaction, but when you look at the total free energy for the binding of the intact peptide, which corresponds to the cognate ligand, the physiological cognate ligand for the SH2 domain, you see that the total free energy for that interaction is about 40 kilojoules per mole. So the phosphotyrosine moiety itself, if you go along with the idea of additivity of free energy, corresponds to something like half the total free energy of that entire cognate ligand or peptide ligand. So it's contributing a significant amount to the free energy of binding. So as I said, and as I'd like to emphasize, trying to replace that phosphotyrosine is a major task. So, as I also said, we decided to try this as an academic exercise by looking at a fragment library, and our approach to this was to use an in silico methodology to start with, where we screened something like 13,000 fragments against the structure of the SH2 domain. And by scoring these fragments as they were docked into the SH2 domain, we could get a kind of league table of which fragments were likely to be better interactors with that PY plus, the phosphotyrosine pocket. So as you can see in the sort of itinerary of how we did the experiment, we managed to pare down the 13,000 fragments to about 14 compounds, which we were then able to test. And we used a method based on fluorine 19 NMR, where we had a SPI, which was a SPI molecule, which was labeled with fluorine, which we could then displace with our molecules in the NMR tube and see whether we were actually getting binding. And this is all, the details of these experiments are all available in the reference that is provided in this slide. So in this slide you can see the data that we obtained for binding of our best hit molecule, and you can see the molecule which is shown below. And the top of the table are the data for the entire peptide, which this molecule clearly, once the fragment was developed into a lead compound, would have to overcome. So the binding constant, or rather the dissociation constant for the peptide is of the order of 0.1 micromolar. And you can see that our hit molecule is clearly significantly less than that by about 3 orders of magnitude. But of course that's what you'd expect. The hit molecule is only a fragment and is only exploring the small surface of that phosphotyrosine pocket. The important part here is that the enthalpic contribution to binding of the hit molecule is actually extremely favorable in terms of the hit molecule itself corresponds to an enthalpy which is more than half the enthalpy obtained for the whole of the peptide ligand. So the question is this a good hit? Well, obviously, in terms of the affinity and the free energy, one would question. It's hard to make a decision as to whether the development of this hit is going to provide a molecule which will compete with the physiological ligand. However, from the point I was making earlier with respect to enthalpic efficiency, this looks like an extremely good ligand, since more than half the enthalpic contribution of the total cognate ligand which we're trying to replace is represented by the hit molecule. And by way of comparison, I can actually show you the data for the SPI molecule, and you can see here that although we've got an affinity which is fairly similar to the hit, the enthalpic contribution is actually unfavorable, and quite clearly the SPI molecule would not be one that we would consider worthwhile taking forward as a lead. So from a fragment-based approach, hits that have favorable enthalpic contribution would be the ones that we would develop. And furthermore, I can show you why this is a good hit molecule. When we actually get a value for that enthalpic efficiency, the hit molecule has a value which is identical to the peptide itself. And so the idea would be then you could actually develop the molecule out and pick up even more enthalpy from additional bonds that you might be able to capture as you build a larger molecule. So I would suggest that in this case, this hit molecule would be one worth pursuing as if you're going to build out your molecule from that phosphotyrosine binding pocket. Clearly, one shouldn't lose sight of the fact that enthalpic efficiency is only one term which can be added into the general mix of decision-making values that pharmaceutical development has to consider. But nonetheless, the idea that you're getting a readout from a simple isothermal titration calorimetric experiment, which can be used to help to establish that there is a good complement of non-covalent interactions in your target ligand interface is a value which I think should be considered closely. Just going back to where the enthalpic contribution from our hit molecule comes from, we can represent the structure of the SH2 domain phosphotyrosine binding pocket and superimpose the binding of the hit molecule on top of that. And you can see in this figure, which is shown in the slide, that the phosphotyrosine moiety in the SH2 domain binding pocket is shown in green here, and the hit molecule is shown in yellow. And you can see the hit molecule adopts a very similar conformation to that of the phosphotyrosine moiety. And so the question becomes, where is it getting its improved or its good enthalpic efficiency from? And when we look at this structure in more detail, you can see, particularly in the representations of binding in the lower portion of this slide, notified by B and C, we've represented the hit molecule binding, which is picking up additional hydrogen bonds through binding to a histidine residue in the binding pocket, and we think that there's additional hydrogen bonds either directly through direct binding with this histidine residue or through the incorporation of a water molecule, which is going to provide additional enthalpic contribution. So the idea of using enthalpic efficiency to look at not only the larger molecular weight compounds but also fragment compounds, I think is something which is worth considering in the drug development process. So the final part of my talk, I'm going to just highlight another area where isothermal titration calorimetric elucidation of thermodynamic parameters can be of use. And this is the characterization of the role of water molecules in a binding interface. And again, I'm going to continue to focus on the interaction of the SH2 domain and show you how the water molecules in the interface of binding of peptides give a clear signature that they're there, and which is an important piece of information to know if you are developing compounds which are going to compete with cognate ligands. So what I'm showing you in this slide is the data for the binding of a series of peptides to the SARC-SH2 domain. And you can see here that the peptides consist of the phosphotyrosine moiety, and then the glutamates, which you saw, and then a residue in the py+3 position. The specific, or the so-called specific ligand, has that isoleucine residue, which I showed you delved into that py+3 pocket. But what we did is we modified the peptide by putting in different side chains. In fact, we changed the isoleucine for all the 20 or 19 remaining amino acids. And here's a selection of those data with different amino acids in that py+3 position. And so we've kept the glutamates as they were and just substituted the py+3 isoleucine. And the isothermal titration calorimetric data is shown in the table here. And you can see that across that range, which is actually somewhat surprising and maybe sort of highlights the fact that, or rather emphasizes the fact that this SH2 domain was actually a major problem to develop compounds against, because if you look at changing from an isoleucine through to different aliphatic hydrophobic groups to aliphatic acid groups, and even large bulky hydrophobic groups, you see nothing more than about a fourfold difference in affinity, which should, clearly shows that that py+3 pocket is quite promiscuous, as you probably would have guessed looking back at those 6 compounds which I described earlier with the different chemical moieties that were supposed to probe that pocket. So the thermodynamic parameters across the board are somewhat similar in as much as the enthalpy terms are all rather large. The lowest is the aspartate group, and the highest is the cognate isoleucine group in that py+3 position. I've also shown on this slide in red the data for another peptide whereby the sequence has been essentially completely randomized. So we've lost our 2 glutamate residues proximal to the phosphotyrosine. And you can see here that there's been a fairly dramatic reduction in the KD. And furthermore, and what is important for what I want to talk to you about in the remaining part of this talk, the enthalpic contribution has dropped off significantly as well. So compared to the cognate ligand with the py and 2 glutamates and isoleucine, you've lost a significant amount, something like a threefold change in enthalpy. So the question is where is this enthalpic? What's the source of this loss in affinity and this clear drop in enthalpic contribution? So when you look at the high resolution structure of the SH2 domain, and particularly in the region where those glutamate residues splay across the beta sheet, which you can see in this particular slide here, you can see that the glutamate in the +2 position, I should actually highlight what you're seeing here. So in the cyan color in the ribbon representation is the surface of the SH2 domain, and the peptide, or at least a short portion of it, which corresponds to the glutamate, 2 glutamates and the isoleucine residue, are shown in the green stick model. And you can see quite clearly that the glutamate in the +2 position sustains a network of 4 water molecules, which are represented by the yellow shapes which interface between the SH2 domain and the peptide. So there are 4 water molecules that form an intricate network of hydrogen bonds between the SH2 domain and the peptide. And as I said, they're sustained by the glutamate in the +2 position side chain. And the numbering that you see on these water molecules corresponds to the order of the strength of interaction that those water molecules are making in the interface, as judged by Leonard Jones and electrostatic potentials. So the question that then becomes is what happens if we modify the peptide and what happens to this network of water molecules? And so rather than going and solving high resolution structures of the SH2 domain with different peptides bound, which clearly is a laborious and difficult task, we thought we'd look for a quicker way of approaching this. So we use mass spectral analysis. And you can see in the slide in front of you now the different mass spectra for the different forms of the SH2 domain or peptide. So at the top there you can see the mass spectra for the apo SH2 domain, the unbound SH2 domain. Below that is the mass spectrum for the PYEEI peptide. Below that, you see the spectrum for the SH2 domain and the PYEEI peptide, and below that you see the SH2 domain with a randomized peptide bound. But of course, the important thing here is to assess what the water content of the protein-ligand interface. And by using a nanoflow system with a electrospray, you can actually identify the bound waters. And if we take the spectrum for the SH2 domain with the PYEEI peptide bound and blow that up so you can see what I mean, you can see very clearly here that if you look at the molecular weight region which corresponds to the protein peptide complex, you can see that there are 4 distinct peaks that emerge from the mass once the system has flown through the electrospray system. The major peak is the SH2 domain and peptide which are bound, but then proximal to that you have 3 other peaks highlighted here by the letters G, H and I. And these correspond to molecular weights which are 18 higher than the SH2 domain peptide. And so these represent peaks which correspond to the peptide with 1, 2 and 3 water molecules bound. As you can see here that the highest population is based on is where you have 3 water molecules bound. If you remember the slide I showed you with the SH2 domain and the 4 water molecules in the interface, the 3 of those water molecules were making a very tight arrangement or network of hydrogen bonds between each other and the interface. The fourth one, which was numbered 4, as I said, based on the strength of interaction, was forming a lesser role in that network. And so what we think is happening here is that you're picking up those 3 tightly bound water molecules, and you see it's a higher probability of seeing 3 water molecules bound in that interface. So we can now use this approach to look at all the SH2 domain peptide binding interactions, and that's shown in the histogram in this slide here. And so what I've done here is take those mass spectral data, which give you a semi-quantitative output of the probability of finding a given number of water molecules in the interface. And I've color-coded the peaks according to having 1, 2, 3, 4, or indeed 5 water molecules bound. And you can see for the most part for the peptides where we modified the PY+3 residue that the predominant or the highest probability is that you'll find 3 water molecules bound. And so that network seems to be sustained across the board. The last one of those, which was the aspartate residue in the PY+3 position, it's kind of a close run thing between having 1 water molecule or 3 water molecules bound. But nonetheless, the trend there for those 5 different peptides was that you would have 3 water molecules bound. So in all of those cases where that glutamate residue is present, you're able to sustain that 3 water molecule network, and that's we can assume is contributing to that similar enthalpy term, because they're all sustaining the network of hydrogen bonds which is required to keep them all in position. However, when we put in a randomized peptide, in this case we have this QPG sequence, you can see quite clearly that the sustaining of the 3 water molecules is actually completely knocked out, and you see that the green peak on the histogram, or the green bar on the histogram, is the lowest, and in fact you have a much higher probability of only maintaining 1 water molecule in that interface. And that's reflected in the thermodynamic data, in as much as you see a significantly lower enthalpic term, as you'll see in the next slide. So for a randomized peptide sequence where you knock out that glutamate in the +2 position and subsequently destroy the water network, you see a significantly less favorable enthalpic term, because you've lost all the hydrogen bonds which are making up that network of water molecules. And by way of highlighting the fact that the thermodynamic data can give you a signature for water molecules being bound, you also see the corresponding entropic contribution for removing those water molecules. So if you look at the PYEEX series, you see that the entropic term is significantly less favorable than the randomized peptide, and the reason for that is that you're trapping water molecules in the case of the or having when you have the glutamate in the +2 position, the water molecule network is sustained. So you're paying an entropic cost for keeping those water molecules in place. And so when you remove that network, you gain an entropic contribution. And so the point being here is that the isothermal titration calorimetric data is giving you a signature of having water molecules in the interface, and quite clearly, if you go through a series, you could get to the point where you can predict ideally predict that the enthalpy and entropy terms are highlighting the positioning of water molecules in the interface. One other thing to bear in mind here is that the enthalpic term is in the case of where you have the water molecules in place is extremely strong and is the major driver of the free energy of binding in this case. And the reason for that is that although you're paying an entropic cost for keeping those water molecules in the interface, you're gaining significantly from the large amount of hydrogen bonds that you're forming through that network. And so what this highlights is in many cases where water molecules are in the binding interface, it's worth looking at sustaining the water molecule network, because you might actually be gaining a significant amount of free energy or favorable free energy from that water molecule network. And so the principle there is that in many cases, the dehydration through development of compounds which remove water molecules actually may not be the way to go in all cases. And it might be worth, in some cases, sustaining water molecules in the interface and gaining from the enthalpic contribution from the hydrogen bonds that they can form, as long as, of course, that enthalpic contribution overrides or overcomes the entropic cost of pinning that water molecule or those water molecules down. And in fact, I can exemplify that in the next series of slides, where we're looking again at a couple of compounds in this case which were developed to be directed at inhibiting the SH2 domain binding. So here again you see two compounds which are peptidomimetic. They both have the phosphotyrosine moiety, which you can see in the bottom, or rather on the left-hand side of the slide. There's a peptidomimetic background, which in this case is rigidified through having a single aromatic cyclic group. And then extending through the molecule to the right-hand side of the slide, you can see two aromatic groups which were designed to probe into that hydrophobic pocket which you saw earlier on the SH2 domain, the PY+3 pocket. So again, these compounds were developed and rationally designed to replace the cognate ligand, which is a peptide, phosphotyrosine-containing peptide, and exploit the two deep pockets that you saw in the earlier slides. One thing that is highlighted on this slide is the positioning within the peptidomimetic sequence of the glutamate in the +1 and +2 positions. And as you saw in the earlier slide, the glutamate in the +2 position is fundamental to sustaining that water network between the peptide and the SH2 domain. And as you can see, both of these compounds have nothing which would replace that glutamate side chain moiety. And in fact, the rationale here was that what the drug designer wanted to do here was to remove the water molecules from the interface and gain an entropic contribution for doing so. So if that was the aim of the process, the drug designer, the drug design team was successful, and you can see that from this superposition of a series of structures of ligands, of peptide ligands, and our lead compound, or one of the lead compounds, bound to the SH2 domain. And I should point out here that in the bottom left of this structural representation is the superposition of the different SH2 domains, which you can see superimposed extremely well, as you'd expect. And then at the top of the slide are the different structures of different peptides and our drug. And I've highlighted the PYEEI peptide, which is in the lighter green, and the drug, which is in the sort of darker cyan colour. and you can see that phosphotyrosine moiety of the drug, which is pointing down. But the important point here is to highlight the water molecules that are either there or not. And you can see in the case of the peptides, the different peptides, which are the water molecules are color-coded according to the peptides that are bound. So you can see water molecules associated with different peptides, and our particular, the one I've highlighted, the PYEEI, you can see a light green water molecule. And I've put a ring around the positioning where you'd expect to see those water molecules, which I've highlighted previously, and numbered those as I had previously based on strength of interaction in the interface, 1, 2, and 3 in this case. The important point, as I mentioned before, is that the drug compound, or the lead compound, completely removes those water molecules from the interface. So you can't see a sort of darker cyan-coloured water molecule in those 1, 2, and 3 positions, suggesting that the interface across where the glutamates would be binding is completely dehydrated in the lead compound interactions. So what effect does this have on the thermodynamic parameters of the interaction? So again, using isothermal titration calorimetric data, you can see that the peptide data shows a tight binding with a strongly favourable enthalpic contribution. As I mentioned, some of that contribution is derived from those water molecules. When we put in the two compounds, which I've labelled DAH1 and DAH2, you can see firstly that the affinity and hence the free energy has significantly changed, that they're significantly weaker than the peptide that they're aiming to inhibit the binding of. But the most important thing here is to reconcile the underlying enthalpic and entropic terms. And you can see quite clearly that we've, by dehydrating the surface, we've gained a significant amount of entropy, which is what the rational approach aimed to do. The entropy term has gone up from about 1.4 for the peptide, to 7 and 8 for the two lead compound inhibitors. But the clear issue that I've highlighted in the last part of my talk here is that by knocking out those water molecules, you've lost a significant amount of enthalpy. And so the enthalpy terms of both of those compounds have become, compared to the peptide, have become significantly weaker than, or significantly less favourable than for the peptide itself. So the point here is that by destroying that network of hydrogen bonds sustaining the water molecules in the interface, you've completely you've lost the enthalpic term, and you've, as a result, got a significantly weaker interaction in the interface. So one offshoot of this particular design strategy might have been that we should try and retain a chemical side chain or chemical moiety that might help to sustain all or some of those water molecules in the interface and provide an enthalpic contribution to binding. So to summarise this last part of the talk, I think it's important to highlight that the water molecules have a dramatic effect on the thermodynamics of interaction, and quite clearly using data derived from isothermal titration calorimetry gives you the only way of directly assessing this effect. The water molecules can be included, and should in fact be included in the understanding of the thermodynamics of the interaction. And again, we need to assess whether a water molecule in an interface is providing a favourable or net unfavourable contribution to the free energy. And I think this is something which is a challenge to the pharmaceutical industry, but nonetheless something which is extremely important. And I think I could emphasise that by the fact that when you disrupt water networks or even single water molecules, you can have a dramatic effect on the free energy of binding. And it's quite, to sort of highlight that, the last point I want to make on the water molecules in an interface is that the water molecules can have a favourable effect on the total free energy, because although you pay an entropic cost for putting the water molecule into the interface, you can gain a significant amount from forming optimal hydrogen bonds from the water reaching out and binding to both the target and indeed the lead compound or drug itself. So to finish, I'd just like to summarize what I've said today. I think it's very clear from the examples I've given that thermodynamic data provides an underlying or an additional level of understanding for the formation of protein-ligand interfaces. And therefore, I strongly believe that it should be data that can be folded into the decision-making process in developing lead compounds. And furthermore, and finally, thermodynamic data provides a signature for underlying effects that can occur in protein-ligand interactions, such as the sequestration of water molecules into the interface. And so having those data has to be of value to the rational drug design process. And at that point, I'd like to thank you for your attention and thank you, Dial, for introducing me.
Thank you, John. A recorded version of this webinar will be archived and available online through the TA Instruments website.